Hey guys, this is Lackey. We are going to be starting on our blood unit. Hopefully you've watched some of the other intro videos I posted for you guys. Um, but we are not going to be doing vocab quizzes. But I would suggest going through the study guide. I have a couple of activities laid out for you guys that'll match what we're doing in the videos for each day. And I am going to have you guys still take the test from home. So I'll have them set up to be open on a certain day. So it'll review all of the material that's throughout the different videos. First thing we're going to start off talking with blood is we're going to talk a little bit about when blood's found at the crime scene, what we actually look for, how we test it, things like that. So whenever blood's found at the crime scene, Um, most often you're going to see this in more violent crimes like fights, murders, assaults, um, possibly break-ins if there was broken glass, things like that. Investigators can use the location, distribution, and pattern of the blood and blood stains to actually reconstruct a crime. Figure out where that blood came from. Is it from different people? Um, if it was from an attack, where exactly did that attack take place? You can possibly link it to a certain type of weapon, things like that. Looking at blood's general biological characteristics, these are all going to be considered class evidence. So just looking at the blood itself, what pattern it is, blood type, things like that are all going to be class evidence. So remember, they're going to lead to a group of individuals. Um, Anytime that there's a transfer of blood between the victim, suspect, and crime scene. And I apologize, I'm having to use my iPad to work and sometimes there's a delay. The only time blood is going to be considered individual evidence is if we're actually looking at the DNA itself. So looking at the base pairs, things like that. Anything other than analyzing the actual DNA, looking at blood type, things like that are all class evidence. Whenever we're at a crime scene and we find what we believe is blood, we have to test it to make sure that it actually really is blood. Because there are some things that may look like blood, but not be blood. So we actually have to run several presumptive tests to confirm the presence of blood. So remember we talked about presumptive tests in our toxicology unit, looking at what things, um, there's a test, normally a color change, that shows that blood should be present. So it shows that blood might be present, but we can end up with a false positive. Just remember, false positives can be from things like bleach, plastic residues, potatoes, and certain metals like iron, copper, and cobalt. So just because that presumptive test shows up positive does not mean 100% that blood is present. So a lot of chemical blood tests can be done as those presumptive tests. All of these are going to react with the hemoglobin in blood. So hemoglobin is the oxygen carrier that is what gives red blood cells their color. But yet again, these tests are still considered presumptive because those chemicals may react with other substances as well. Even if you don't have fresh blood, if it's dried, stained, anything like that, you can still test it for blood. So a lot of blood stains will be tested with luminol. So after it's been attempted to clean up, it's been dried, things like that. Luminol is a very sensitive chemical indicator for dried or washed blood, and it causes chemiluminescence. Chemiluminescence is the emission of light or glowing from a chemical reaction. So you may have seen this in crime shows, especially if they think the crime scene's been cleaned up. They'll spray a chemical, a lot of times use a UV light or something like that, and it will glow a pink or purplish color. This is similar to what happens in fireflies or lightning bugs, which is the bioluminescence. It's the reaction within their body. Once we say, okay, this is likely to be blood, we have to figure out, is it human blood, animal blood, or yet again, is it not blood at all? So once it's determined that a stain is blood, we have to figure out the species using a Preston test. A Preston test will distinguish between human and animal blood. So normally a Preston test contains an animal serum that contains antibodies specific to human antigens. So the antibodies will attack any human antigens and cause clumping if human blood is present. So the animal serum is a liquid that's separated from clotted blood. So it still has parts of the blood. It will not clot itself. It just contains all of those antibodies and antigens in there. The antibodies, remember, are proteins in blood or any other secretory fluids like saliva or sweat that are going to tag, destroy, or neutralize bacteria, viruses, or other harmful toxins, producing an immune response. The immune response we look for is clotting. Antigens are foreign substances in the body that are capable of causing disease. So these could be any type of infection, bacteria, things like that. So if these are present, they trigger an immune response in the body, which produces antibodies, and the antibodies will attack those foreign antigens. The serum is going to react with the antibodies to cause agglutination in human blood. So agglutination is an allergic reaction, which is going to be seen as clumping. So the red blood cells are going to end up clumping together, usually in response to a certain antibody being present in that serum. So anytime that you're testing for blood type and you see clumping, it means that that blood type is present or that blood antigen's present. We'll talk about that in a little bit. These tests are so sensitive that even if blood has been diluted, it's rained and washed away some of it, or it's on a really, really old dried piece of evidence, you can still test for blood and blood type, things like that. These Preston tests can also use anti-serum, which will contain the antibodies instead of specific antigens. Um, Looking at bodily fluids in blood itself, so serology is the study of bodily fluids. Um, this includes every bodily fluid, but a lot of it is looking at blood. About 1/12 of the human body is completely blood, and blood is not one consistent substance. It is going to be a mixture with different parts. The fluid part of blood, which is our plasma, makes up about 55% of all of our blood. Plasma is going to be about 90% water and 10% metabolites, waste, salt ions, and proteins. Metabolites are organic molecules involved in process of metabolism. A lot of times these are things that are being filtered throughout your body and broken down, like ketones, proteins, things like that. Um, with the salt ions and stuff, the most common ions you're going to find are sodium, chloride, stuff like calcium, or hydrogen carbonate. The more solid part of your blood, so that other 45%, is made up of three types of cells. We have our erythrocytes, which are red cells. This is where you're going to find that hemoglobin. The whole purpose of hemoglobin is it is to transport oxygen from the lungs to other cells in the body, and then it carries that carbon dioxide back to the lungs to be exhaled. We have our leukocytes, or white cells. These are our cells of our immune system that produce those antibodies. And then we have our platelets. Platelets are used for clotting. So it starts the clotting process by initiating the formation of fibrin. And that clotted material is what's used to make the serums for different blood tests. In 1901, Austrian biologist Karl Lansinger recognized that all human blood is not the same. They noticed that when they were trying to give people blood, they were having adverse reactions and dying all of a sudden. So this led to the development of our ABO classification system that we still use today. Lansinger also later developed the rhesus factor, RH factor, in blood. This you know as the positive or negative with the different blood types. And it led to the development of over 100 different factors in blood. Um, so not all blood is the same. Even if it's the same blood type, there can still be different proteins and factors in that blood that might be useful in different types of transfusions. Theoretically, no two people have the same blood except for identical twins. So identical twins should have the same blood factors. Um, this is just a different combination of antigens, enzymes, and proteins in the blood. So it may be someone who's going through chemotherapy that needs a blood transfusion needs blood with certain antigens in it and certain blood factors in it to help them refresh whatever they've lost. A lot of these blood factors are looked at for blood typing because individual blood analysis is time consuming and expensive. So it's more likely that we're going to test for blood factors or blood type than DNA just for time and money's sake. The ABO and RH blood characterization is really important because it can be done with whole blood or dried blood. See, yet again, even if we have an old dried blood sample, we can still find that blood type itself. And about 80% of the population are what we consider to be secretors. They actually also produce these antigens in other bodily fluids like urine, sweat, saliva, semen, things like that. So looking at the blood cells themselves, the antigens that are actually on the blood cells are responsible for the different blood types. So we have A antigens and B antigens that will determine what your blood type is. The white blood cells manufacture the antibodies, and these attack any invaders in your bloodstream, like any foreign antigens that should not be there. Venom, if you get a snake bite or something like that, they can attack that venom itself. Bacteria, infection, or someone else's blood. So here's what it looks like if you have different blood types. If you have type A blood, that means that your red blood cells contain A antigens. So the antigens are what gives it the blood type. And your white blood cells will produce antibodies for type B. So you will attack any B antigens that enter into your body. If you have type B blood, your red blood cells have B antigens on them, and your white blood cells will produce A antibodies. If you're type AB, your red blood cells are going to have both A antigens and B antigens, and your white blood cells are not going to produce antibodies for either of those antigens. Whereas I'm type O, so my red blood cells do not have any type of antigen on them. But my white blood cells are going to produce both A antibodies and B antibodies. So where this gets used a lot is with blood transfusions and blood typing. Antibodies are what we consider to be bivalent, so they essentially have two sides. So they can attach to two different antigen sites, which causes the red blood cells to clump together, which is that immune response we look for. So if a person with type B blood receives a transfusion with type A blood, their A antibodies are going to attach to those blood cells with the A antigens and cause clumps, which can move throughout the bloodstream, cause strokes, things like that, and become fatal. This is why blood typing is so important before they do a blood transfusion. They're not going to give someone any type of blood unless they know their blood type. Um, and they can actually do transfusions with plasma and whole blood. So they have to match both of these with an appropriate blood type. Here's kind of how they know who can receive blood and who can donate blood. So remember, if you have type A blood, you have A antigens and B antibodies. So you can donate to someone else who is not going to interfere with your A antigens. So you can donate to someone else who is also A, or someone that is AB, because they still have A antigens and no A antibodies. But you can only receive blood from A blood because it will not have A antibodies, and O blood because your B antibody is not going to interact with anything on O. If your blood type is B, you have B antigens and A antibodies. You can give blood to someone else who is B or someone else who is AB because their antibodies are not going to interfere with your B antigens. But you can only receive blood from someone who's B or someone who's O because you cannot receive A antigens in any form. If you have AB blood, you have both A and B antigens and no antibodies for blood type. You can only donate to someone else who is AB because you have both antigens. However, you're what we consider to be the universal recipient because your antibodies are not going to attack any type of blood that you are given. Whereas if you have O blood, you don't have any antigens present, but you have antibodies for both A and B blood. So you're what we call the universal donor. Anyone can take your blood because there are no antigens on the blood cells, but you can only receive blood from someone else who is also O. Another part of blood typing is that rhesus factor, Rh factor. This is important, and sometimes it's going to be referred to as the D antigen. So the positive means that you have this antigen. The negative means that you do not have this antigen. So people with the antigen are Rh positive. This is going to be about 85% of the population. If you do not have this antigen, you're Rh negative. And we have a table here of the approximate percentages, but this distribution might be different. You might have areas with more A than O, things like that. I'm actually missing a part of my table. There we go. So if you notice, the most popular blood type is actually O, with about 45% of the population, followed by A, which is about 40% of the population, then B, which is 11, and the rarest blood type is AB, which is only about 4% of the population. Majority of people do have that D antigen, so they are considered positive, whereas about 15% of the population are negative for that antigen. Using all of this, we can look at probability to figure out what percentage is A positive, A negative, things like that. So blood typing can show how two blood samples have different origins. So most of the time, blood typing is due to exclude suspects versus prove someone's guilt, because yet again, this is still considered class evidence. It is not going to point to one person. The distribution of blood types in a population can be used to calculate the probability of a suspect having a certain blood type. So if you cannot test for DNA and all you have is blood type, this is kind of how you would present in court of what percentage of people in that area are going to have that same blood type. So we did probability back in Unit 2. It's been a while. But remember, you end up multiplying the percentages together to get the combined percentage. So if we want to know what part of the population would be positive, we would have to multiply those two percentages together. So remember, if someone had B blood, that was 11% of the population, so that's 0.11, times the people that are positive for the Rh factor, that was about 85%. So if we do 0.11 times 0.85, that is 0.0935, which is about 9.4% of the population. So say that we were in a town of 1,000 people. That would be approximately 94 people with this blood type. So that's how you can present this in a court of law. Let's look at our rarest blood type, which is AB negative. So AB negative is the rarest, O positive is the most common. So if we wanted to look at the probability of someone being AB negative, remember the percentage for AB was 4%, so 0.04. The negative was about 15% of the population, so 0.15. That's going to give you 0.006, which if you change into a percentage is 0.6%. So if we were in our same town of 1,000 people, only 6 are likely to be AB negative. So we can use that to narrow down our suspect field. Blood can be tested for type by mixing a drop of blood with a drop of that serum or antiserum that contains known antibodies. If that agglutination, if clumping occurs, you know that type of blood or that type of antigen is present. Whenever you're testing dried blood cells, the red blood cells have actually ruptured, but those antigens and antibodies are still going to be present. And remember, when packaging any blood evidence, you don't want anything wet to go in a sealed container. So you want to make sure it's in a breathable area, that you give it a chance to dry to prevent mold and mildew. You're going to be blood typing in the activity, the blood typing game, and we'll do a little bit more with blood spatter tomorrow. Thanks, I'll see you later.